Welcome to the ACUR ANU student presentation session. Firstly, I'd like to acknowledge and celebrate the first Australians on whose traditional lands we meet. So for me, it's the Ngunnawal Ngambri people here in Canberra. I want to pay my respects to their elders past and present. So my name is Asha Samaris and I'll be chairing today's session. Our first presenter is Callum Ormonde, and please take it, Callum, when you're ready to start.
Sure thing. Thank you, Asha. And just share screen. All right, are you seeing that? Is everyone seeing that?
Yeah, all good, thank you.
Wonderful. Okay, so thank you very much for everyone coming and welcome. My name is Callum Ormonde. I'm a medical student at the University of Wollongong, and my presentation is on perioperative clinical practices in postoperative delirium patients in older people. So just beginning with some definition of terms. So delirium is defined by its acute and reversible nature and its altered level of cognition and attention that results from that. When we see delirium, it presents in three different forms, that being the hyperactive, the hypoactive, and the mixed or combined form. Perioperative care, this is the care for patients both before and after surgery, but doesn't include intraoperative care. And why am I concerned about older people or older patients with regard to postoperative delirium? Well, look, it's because they're particularly at high risk of postoperative delirium. And most of this is attributed to two primary facts. So their loss of cardiac and pulmonary reserve that just happens with time, together with the increased incidence of their comorbidities. So the impact of postoperative delirium is seen where anywhere between 1 in 10 to as high as 1 in 3 elderly people receiving surgery will have postoperative delirium in some form. The consequences of this are many, and I've got them listed here. But what I'd like to particularly draw your attention to is the cognitive impairment that we see in over half of patients, where the impairment isn't just the acute presentation in hospital, but can actually leave patients cognitively impaired for as long as a year afterwards. And as you can imagine, in the context of an aging population, this is a problem that isn't going to improve, is only going to get worse for the foreseeable future. So the management of post-operative delirium is largely left to nurses in hospitals. And this involves a multi-step approach outlined on the slide at the top. And what I've included on the right here is one of the many tools that we use to aid in the management and diagnosis of delirium, the so-called 4 A's test. And this is the sort of bread and butter gold standard that most people on the ward would be using for diagnosis. But despite tools like this one that I've shown on the right and protocols being in place, there are still many problems that exist. So specific things that we notice are often difficulty in detecting, and that's especially true of the hypoactive form, which simply presents as a quieter state. And if you imagine the squeaky wheel gets the attention, quiet one often goes missed. So that's one of the difficulties that occur day to day, as well as misdiagnosis. So elderly patients are often misdiagnosed as simply being demented or experiencing dementia and go untreated for their delirium, as well as another of other practitioner-based problems that are outlined in there, but I'm just going to move on for time. So we set about looking into this problem, and what we aim to do is assess the knowledge and understanding of currently practicing nurses in Australia who are working on surgical delirium care wards. Our objectives were to determine whether the amount of education in postoperative delirium or any form of delirium could be predictive in nurses' responses to ward or case-based responses of postoperative delirium. And what we focused on is nurses' self-reported confidence and competence in the diagnosis and management of postoperative delirium, as well as also looking at things around the issue like risk factors and their performance in case-based scenarios. So we set about designing a survey and distributed that through the SurveyMonkey function via email invitation to hospitals all around Australia. In short, this was 27 questions looking at a number of different demographics and background, education levels that nurses experienced. When actually receiving the data, so the data came back, we received 424 responses and excluded 98 of these for not meeting our inclusion criteria. So the exclusion criteria are shown on the left and the inclusion are on the right, which left us with 336 responses that were either answered in part or completely. We then looked at the responses and respondents' answers to a particular question regarding whether the respondent had had any form of delirium education, be that in their undergraduate, postgraduate, or continued professional development, or whether they'd had none at all. And respondents' answer to that question created 3 groups or cohorts. So our first cohort had no formal education, and this was just over 100 individuals. Our second group had experienced either 1 or 2 previous education sessions on delirium, and they were just over 200 individuals. And our third group identified either 3 or more times in which they've been educated on delirium. The group demographics are also shown on the right here, but once again, for timing, I'm just going to continue on. So with regards to our question of nurses' confidence in the management and treatment of delirium, we asked them to specifically rate their confidence in detecting hyperactive and hypoactive delirium, which are shown in graphs A and B. And then we asked about their confidence in management of the 2 forms, shown in C and D. And this was done on a sliding scale from no confidence to highly confident. And the results that we found that across all 4 aspects of delirium care, the majority of respondents were rating themselves either slightly or moderately confident in their skills. All groups responded that they felt more confident in detection than management of delirium care. And we noticed statistical significance in confidence levels relating to education in the particular hyperactive form of delirium. So what we took this to mean is that nurses with a higher level of delirium education were significantly, excuse me, more likely to feel confident in their ability to detect typical or hyperactive delirium. And we put this down to the more overt nature that hyperactive, agitated, aggressive forms attract more attention, and so it becomes much more obvious to diagnose. Whereas the converse relationship, that nurses didn't feel confident in their ability to either diagnose or treat hypoactive forms, is likely due to it simply being less experienced. With regard to nurses' response in competency in our 4 previous mentioned aspects of delirium care, once again, we noticed that the greatest competence was reported in detecting of hyperactive form. Whereas what we saw in this instance was that there was statistical significance between reported competency and education, not only in detecting hyperactive, but also managing hyperactive delirium. And this was particularly interesting with regards to, it seemed counterintuitive. The idea that nurses could perceive themselves competent in a skill that they didn't deem themselves confident in was very counterintuitive to us. And it's something that we're continuing to look into at the moment. So when we looked at our responses to case scenarios, in the top left here, we've got responses to risk factors. So we gave our nursing staff 18 risk factors and asked them to say whether they were aware of this before or this was new information. And the general trend that we found was with increasing amounts of previous education, the range of scores was narrowed and the mean average score out of 18 was increased. However, all three of the groups still were able to produce scores of 18 out of 18, perfect scores, which we took to mean that nurses do in fact possess the in-depth theoretical knowledge required for post-operative delirium care, but that this may not be translating to on-the-day actual practical performance, which we observe later when we're looking at these case scenarios. So the case scenario we gave is shown down in our bottom left corner. And we then asked two questions about the immediate care for delirious patients and then protracted care. So question 2 is the protracted care in table 4 and the immediate care, table 3.
Thanks,
So,
Callum. I'll just have to step in there because your time's up.
Okay, thank you.
Thank you, Callum. There's 5 minutes now for questions, and I'll just hand over to our judges if they've got any questions for Callum.
That might save everyone a little bit of time.
Oh, there you go. Am I muted or can you hear me?
No, I've got you now, Barnaby.
Hey, Callum. That was really, really nicely put together, really well-designed slides and really clear. I was just wondering, is the onset of post-operative delirium in its various forms, is that due to reaction to anesthetics, or what's the underlying cause, if it's even known?
Yeah, so the precise pathophysiology is not exactly known, but certainly anesthesia is one of the largest risk factors. So I talked about cardiac and respiratory reserve and comorbidities. Effectively, the two things that are most affected by the anesthesia of the surgery rather than the actual procedure performed. So yeah, that's certainly why it's one of the largest responses. But it's so multifactorial that it's why the prophylaxis and risk identification are 1 and 2 in the management procedures.
Okay, thanks very much.
Darren, did you have any questions?
Okay. Hi, Callum.
Hi.
Yeah, so I would just like to get something clarified. Is in your presentation, you have stated that you measured the nurses, clinical nurses, in terms of confidence and competence. Are these two aspects being measured through the 4A assessment?
Oh no, so they were self-reported via the online survey. So the online survey that we mailed out had both ranked response and sliding scale responses. So we offered them, for example, do you feel confident? Please rank your confidence in detecting hyperactive delirium. And that would be none whatsoever, some, moderate, and highly confident. And it was that ranked score, which then we coded into responses.
So it's a personal assessment kind of thing.
Yeah, this survey data was, yes. The 4As is a tool that is used to try and remove the personal aspect. That's used on the ward in an effort to try and homogenize and protocol detection of delirium.
Right. Thanks for the clarification.
Thank you.
We've got a couple minutes left for questions. So if there are any audience members who would like to ask a question, please put it in the Q&A box, and I'll just hold off for 30 seconds to see if there's any questions.
well, if I've got a minute left, this was my last slide and the slide backgrounds. So I'm in Wollongong and it's week 11 of lockdown. So I've been filling my time with art history. And this is the Wild Hunt of Odin. Odin's name comes from the root for delirious rage.
Oh, wow.
Thank you.
All right.
Okay, thanks so much, Callum.
Thank you very much. All right, I'll just move out of share.
Perfect, thank you.
Thank
So
you.
our next speaker, thanks, Callum. Our next speaker is Max Kirkby. Max, please start when you're ready.
Yeah, no worries. Can you guys see that? Let me just, full screen, is that all good? Okay, perfect. So hi everyone, my name's Max Kirkby. I'm a member of the Clear Vision research team here at the ANU. And this year I was lucky enough to conduct a project looking at the effect of voluntary exercise on light-induced retinal degeneration. So as we all know, exercise has large protective effects for almost all systems and organs throughout the body. And we're constantly reminded of that every day. However, it's actually remained unknown why exercise is conferring this protection. And that was actually the case until recently, when studies showed that muscular contraction upon exercise can release certain types of molecules into systemic circulation. And these molecules can act via diverse pathways, for example, in inflammation and metabolism, to confer the previously identified protection against diseases such as cancer and type 2 diabetes. Now, this was obviously really interesting, and particularly so for our lab group, given that exercise had also displayed importance in the central nervous system. And this was particularly the case in Parkinson's and Alzheimer's, which are two of the most common neurodegenerative conditions. However, while there was a wealth of literature, there was actually sort of a lack of research done on retinal degeneration specifically. And this was quite interesting because, as we know, the retina is actually an extension of the central nervous system and is similarly classified with the brain. So quickly, the retina is basically the light-sensitive neural tissue lining the back of the eye and is responsible for converting light energy that we might see every day into electrical signals that can be understood by the brain. And this occurs through a process known as phototransduction, which is facilitated by cells known as photoreceptors. Unsurprisingly, you might therefore guess, the degeneration of the retina is ultimately characterized by the death of these functional cells known as photoreceptors. And one such degenerative condition is known as age-related macular degeneration and causes the death of photoreceptors in this highly specialized region highlighted here, known as the macula, which confers basically all of our useful vision every day. Interestingly, it's actually a major cause of blindness in the developed world, particularly in people aged over 50, and there's currently no known cure for AMD. So this wealth of literature and exercise, combined with a lack of sort of efficacious therapeutics, led our lab group to sort of postulate whether it might be possible that exercise could actually protect retinal function through some form of mechanism in AMD. And to do this, we used an animal model-based approach where we basically took two sets of mice and either ran them or not on a voluntary treadmill for 4 weeks. Following this period, we basically artificially induced AMD using this toxic bright light at 100,000 lux, which is about 2,000 times brighter than we might experience every day. And this mimics the underlying pathogenic features of AMD. And so what we really wanted to see through this experimental timeline was whether preconditioning these mice with exercise could actually improve or protect retinal function in subsequent AMD, and whether it could actually protect these photoreceptors, these functional cells of the retina. Furthermore, we also wanted to see whether this protection might have been occurring due to changes in inflammation or gene expression within the retina. So knowing all this, we pondered off and sort of established our first aim, which was obviously to validate that voluntary exercise could actually protect retinal function. Because, you know, if we couldn't show that it protected just pure function, the project was largely redundant. And to do this, we used a technique known as electroretinography, which measures the response or the response of the retina when exposed to specific light stimuli and how well these cells respond. And we tested two types of cells initially. And so these were firstly photoreceptors, which remember are these functional units of the retina, but also bipolar cells, which facilitate the communication of this message onto the brain. And in both instances, across all light stimuli, we saw a significant increase in the response of exercised mice when compared to controlled mice. And this sort of led us to suggest that exercise was actually really able to protect retinal function in this photooxidative model of AMD. So next, obviously, we wanted to see whether this improved retinal function was potentially occurring due to some changes in the underlying functional units, these photoreceptors within the retina. And to do this, we collected the eyes from the mice after electroretinography and conducted a fluorescence assay using a tunnel marker, which I'll get to in a moment. And this pretty image here basically shows tunnel labeling, which marks photoreceptors and general cells as they're undergoing cell death. And this process and quantification really allowed us to see the amount of tunnel positive cells and amount of photoreceptor cell death occurring throughout the entire retina. And as we can see here, there's clearly a significant reduction in the amount of photoreceptor cell death that's occurring in these exercised versus controlled mice. So we've established that photoreceptors are clearly being maintained better due to exercise. However, might there also be some other retinal inflammation or inflammatory changes that are occurring? And to do this, we conducted a similar assay, this time labeling for IBA1, which is basically just a marker of how many immune cells in the retina are being activated at any one time. And so as we can see here again, labeling these cells within the retina, we again observed a reduction in immune cell activation in the retina in exercised mice when compared to controls. So clearly, not only is there better maintenance of these photoreceptors, but there's also a reduction in inflammation that might be driving some of these protective retinal function features. However, there's obviously a huge spectrum of things you can investigate in molecular biology. And one of the most important things to investigate is a differential gene expression that can sort of regulate everything we've just previously discussed. And so we really wanted to check this as our sort of final aim within our methodology. And so we took samples from our collaborators at Emory University and basically conducted gene expression analysis using a specific assay from the retinas that we'd previously isolated. And we really wanted to test these specific molecules known as MicroRNAs, which are quantitative regulators of gene expression. So they can basically regulate the expression of genes all over the retina specifically. And this has actually recently been shown to be very important in the survival of these functional cells, these photoreceptors within the retina. And so we tested microRNA 182 and 183, which are part of the photoreceptor cluster, which help to maintain the synapses and various other sort of homeostatic mechanisms of these photoreceptors, but also microRNA 124, which are specifically related to age-related macular degeneration. So we've got these three microRNAs, sorry, and we conducted subsequent gene analysis on these retinal samples. And what we found was that these microRNA, specifically microRNA 182 and microRNA 124, were being significantly upregulated as a result of exercise in the degenerating retina. So exercise was actually increasing the expression of these microRNA. And this was obviously a really interesting result given the multitude of roles that microRNA can play in the retina. So in summary, we've obviously sort of come up with a huge wealth of information throughout this 6-month project. Firstly, we showed that voluntary exercise can in fact protect retinal function during this photooxidative model of AMD. We were then able to show that these exercised mice actually display reduced photoreceptor death and inflammation, which might sort of be mediating the protection that we initially observed. And finally, we also saw that these molecular effects might be being controlled by microRNA in the degenerating retina, which can mediate the transcription and expression of specific molecules within the degenerating retina. So obviously, these are very exciting findings, especially for our lab group. And we think that some of this work can help us to understand the diverse molecular pathways able to mediate neuroprotection, but also, I think most importantly, help to identify molecules able to supplement the degenerating retina. So for example, if you have an elderly patient, which is the case usually with AMD, might it be possible to artificially inject microRNA into the retina, providing the same benefits as exercise without actually doing it, which is obviously everyone's dream. And so in this scenario, we might actually be able to provide finally a non-invasive, potentially inexpensive AMD prevention option for individuals. So obviously none of this work would have been possible without the fantastic help and assistance I've received from Clear Vision Research, and particularly Ricardo Natoli and Joshua Chutan, who have been my supervisors throughout the previous semester. So thank you very much, and I'll open it up for any questions.
Thank you, Max. If our judges have any questions for Max.
Hey, Max.
Hi, Barnaby.
It was a really, really nice presentation, really interesting. I was just wondering if there has been any work looking at just correlational work among aging people who do present with this, with the degeneration of the retina. Is there any correlational evidence that exercise is associated with better retinal function among aging adults?
Yeah, so there have been some longitudinal studies that have actually shown that sort of moderate exercise, obviously self-reported, does reduce the incidence of AMD. However, that's sort of been quite inconsistent in a way, because obviously the amount of exercise that people do varies hugely. And so one of the future aims of our group is to really try to correlate the amount of exercise that someone or a mouse is doing with the ultimate retinal function that they might present with. But yeah, it's definitely been shown in the literature.
Oh, cool. Thank you.
Hi, Max.
Hi, Darren.
I was just a bit curious about your methodology when you had a control group and you have a test group for your mice. So as I understand, your test group, the mice test group, has 4 weeks of voluntary exercise.
Correct.
Correct. So how do you make sure that the mice is actually doing the exercise? Like you do not able to pinpoint exactly how much exercise has been done throughout the 4-week period?
Yeah, so basically we had these specialized treadmills, if you could believe it. And basically we're able to hook these specialized treadmills up to a central computer that logged the distance and the speed run by these mice every time they stepped onto it. And so one of the things that was really important for us was to make sure this was a voluntary model. There have been previous studies in exercise physiology that really shock the mice to run, and therefore sort of like completely standardize how much they run per day. But we sort of ensured we had a voluntary model in which we could log the data to actually see whether the mice were in fact running. So that way we sort of kept tabs on that they were running, but made sure we weren't forcing them to exercise.
So within the 4-week period, approximately how long are the mice actually exercising? So
I'm not sure if you're going to believe this, but on average, every day the mice ran 10 kilometers. So
that's huge.
Yeah, I know. Well, they're really tiny, and the treadmills aren't large, but on average, it was sort of between 10 and 12 kilometers of running per day. And obviously there was some variation. Some mice didn't want to run, some wanted to run a lot. But yeah, on average, it was about 10 kilometers a day.
That's cool. Very interesting study. Thanks, Max.
No worries.
Thank you, Max. I've just got Callum with his hand up. Callum, do you have a question?
Yeah, Max, so you said that that you've noticed that exercise in human models reduces incidence. Is it, do you mean by that, like, is it therapeutic? Like once people have a diagnosis of macular degeneration, if they start running, does it slow or prevent progress?
Yeah, so what the literature has basically looked at is sort of preconditioning. So this is what we've sort of looked at in this study as well, is that people that regularly exercise throughout life or a specific period in which the study has taken place are then less likely to develop AMD. And one thing that is being looked at the moment is, you're correct, this therapeutic option, as in once people do start to develop AMD, can we actually then prescribe exercise afterwards and then use it as a therapeutic option? And so that's something that has been recently explored. There's no real preliminary data, but that's definitely something we also want to test in our model, is that can we give these mice artificially induce AMD and then prescribe the exercise and see similar effects?
Cool, thank you.
No worries.
Thank you. Thanks so much, Max. And I'll just turn to our last presenter now, Vrinda Jain. You can start when you're ready. Just get Max to stop sharing, and you should be able to share your screen now if you've got a presentation.
Can everyone see my screen? Yep, all good. All right, so I just want to say my name is Vrinda and I'm from Western Sydney University, and I've been looking at the development of dopaminergic cells in the mammalian retina. So it's a little bit similar to Max's looking at the retina. I just want to quickly thank my supervisor, Dr. Morgan Cameron, who this project wouldn't be possible without. All right, so before I can begin talking about my project, I'll just give you a bit of the background information running all those fun words. So with the retina, again, Max did a very good job of explaining its function, but it's the cell, it's a layer of cells in our eye that basically transmit and let us collect process visual information and transmit it to our brain. So as Max mentioned, our retinas have photoreceptors. So we've got our rods and cones, which are the main ones. Rods typically look at our vision in low light, and cones typically look at color. We also have a different type of photoreceptor called melanopsin, which isn't shown on this diagram, but they're functional photoreceptors before rods and cones mature, so before birth and around then. Another cell I want to draw your attention to is amacrine cells. So as you can see, you've got this pathway from photoreceptors to ganglion cells. And amacrine cells basically regulate and modulate the pathway between bipolar cells and ganglion cells before that information is delivered to our visual cortex in the brain. So that's just one cell that I'll be looking at in my project. And then, of course, we're looking at some dopaminergic system in our eyes. So most of us know dopamine as the happy chemical, but it actually has a huge role in the central nervous system, and it's involved in a variety of physiological processes, including movement, memory, and things like motivation. In the eye particularly, it's actually released in response to light. And I guess a key significant thing to note with this is any situation, or a lot of situations in which you have dopamine dysfunction, that can play a huge role in diseases like Parkinson's disease, schizophrenia, and ADHD. In the retina specifically, so the dopamine system, or the dopamine circuitry, plays a huge role in light adaptation, the development and modulation of neuronal circuitry, so those cells that we saw earlier, as well as things like eye growth and vision for myopia. And I'm just like putting a little diagram of the dopamine chemical. So what we're trying to do is gain a better understanding of the dopamine system in the retina, given it has such a huge role in like light adaptation and developing that actual neuronal circuitry. And surprisingly, given the fact that it is so important, we actually don't really know a lot about the mechanism behind how it's released. So we're taking a look at some of the main dopaminergic cells in the retina, known as dopaminergic amacrine cells. So those amacrine cells I was talking about earlier. And the ones that we're looking at in particular are called TH cells. So just to clarify, TH cells are a type of dopaminergic amacrine cell and are a huge part of your dopaminergic system. So how do we get about this? We collected a wide variety of retina samples across development of the mice. So we looked at postnatal days, so after birth, so 2 to 3 days after birth, 6 days, 8 days, 14 days, 19 days, 4 months, and finally an adult retina or an slightly aged retina. We looked at these retina samples exposed to both light and dark, and we looked at them as a whole retina as well as cross sections of the retina. So one key thing to note is for mice in particular, eye opening occurs around 12 to 13 days. So 14 days is right after eye opening, and P8 or 8 days is before eye opening. And just to say, rods and cones are typically more active after eye opening. With our actual method, so we stain for our TH cells and as well as CFOS. So TH, so whatever comes up green on the next couple of slides is going to be our TH cells, and everything that comes up in red is our CFOS, and that's a marker of neuronal activity or depolarization, or in this case, our dopamine release. So what did we find with this project? So if you guys want to take a look at these slides, you can see these two images. So these are, again, our retinas. So this is an adult sample. So this one's been exposed to light. Can you guys see my mouse? Okay, awesome. So this one's been exposed to light and this one was left in the dark. So it's a little bit hard to appreciate from this diagram, but you can see how there's a little bit of red inside these TH cells here. So we've got this CFOS expression inside our TH cells in the light retinas, but not in the dark retinas. So from that, we can see there is that kind of neuronal activity happening in these TH cells when they're exposed to light. So a similar thing we can see with our P14 or 14-day-old mice samples. So this is a slice or a cross-section of the retina just to give us a better idea of cells themselves. So again, you can see in the light exposed retina samples, there's got that CFOS expression within our TH cells, but not in this dark sample here. And you can also see that this TH CFOS expression in our TH cells actually happens in 100% of our TH cells across the adult and the P14 retinas, which is a bit hard to appreciate with this cross-section, but that's that story. Now, interestingly, this doesn't happen in our P8 samples, so 8 days after birth and right before eye opening. So you can see that even though we have a little bit of CFOS expression, there is zero overlap between our CFOS and our TH cells. So we're not getting that TH activity, we're not getting that dopamine release from our TH amacrine cells. And again, in the dark, it's the same thing. There's no expression of CFOS, there's no activity, there's no dopamine release. So what does that mean? We can see that CFOS, so TH cells are definitely activated in response to light, but only after that stage of eye opening. And as we were talking about earlier with our rods and cones being a bit more active after eye opening and melanopsin being the main photoreceptor that's active like around birth and up until rods and cone maturation, we can assume that these are likely going to be rod and cone driven signals. So it's probably the rod and cone photoreceptors that are driving this kind of dopamine release in response to light. In response to light. What else we found is, again, so looking at different age groups. So it's a little bit hard, again, to see on these diagrams, but we looked at the density of our TH cells and our CFOS. So we counted all of the cells and we looked at how that changed across development. So again, it's a little bit hard to see because the P8 TH cells are a little bit smaller and a little bit dimmer, but it's actually a decrease in density of our TH cells or our green cells across age from P8 up until adulthood. But the opposite actually happens with CFOS. So it's a little bit, again, hard to see, but you might be able to appreciate that the image themselves is getting a little bit redder. So a lot more CFOS expression as we go on from P8 up until adult. So this is just a graph to show the kind of quantification of the averages of our TH cell densities and our CFOS cell densities. So we can see after day 8, we've got this kind of decrease up until adulthood of our TH cell densities, but the complete opposite trend for our CFOS, we've got this kind of increase in our CFOS expression. So that's kind of what we can see here. And again, so that kind of signal after eye opening. The last thing that we looked at was a little bit about distribution of our TH cells and across the retina. So you can see in both these diagrams that there aren't really any clusters or there's no TH cells like kind of gathering in one region of the retina. It's sort of spread throughout the entire retina. It's pretty even. So that's kind of what we saw. There's no variations in cell distribution. You know, we marked these cells for kind of where the dorsal side is. So for both of these retinas, the dorsal side or the top half of our retinal section is at the top and the ventral side or the bottom half is at the bottom here. So again, there's not really any variations. There's no clusters of TH cells at the top or at the bottom. That was our final finding. So I guess the key take-home message to point out is that, you know, we've got this kind of rod and cone-driven release of dopamine in our TH cells after that eye opening, and that increase in neuronal activity after that eye opening as well. So I guess where are we going to go from here? So this has given us a little bit of an understanding about what drives dopamine release in development of our retina. So going on from here, maybe looking at aging retinal samples. So we've gone up until about 4 months. If we can go a little bit further and see how degradation or age might influence dopamine release in the retina. We also want to look at cell inputs into our TH cells. So again, we can assume that it's going to be like a rod and cone driven input, but exactly how that's happening is something that we want to look further into. And then finally, looking at the quantification of dopamine release. So while we can say that dopamine is being released, the actual quantity of dopamine is still some work we need to do, and try and see if that changes across development at all. So thank you everyone for listening. That's my presentation, and I welcome any questions.
Thank you, Vrinda. I'll hand over to our judges again.
That was a really nice talk. Thanks very much. You were saying it's important to look at your last point. I was going to ask something about quantities of dopamine and their influence on this process of retinal development. What would you expect to happen? Have you got any hypotheses there regarding how the quantity of dopamine and what it may or may not do?
So it's a little bit hard to tell, but for TH is actually an enzyme in the dopamine production pathway, so it's tyrosine hydroxylase. So when we had P8, if you remember, the cells were actually a little bit dimmer, so that could be less TH. We could expect less dopamine being produced as compared to the samples in your adulthood. So I personally would expect a kind of increasing trend in dopamine release, but again, it's hard to tell without the actual data.
Yeah, no, that's fair enough. Yeah, that's exciting work. Thank you very much.
Thank you.
Hi, Vrinda. Hi, I just want to ask, like, as I understand from your study, you are doing your study based on a mice model similar to Max. So how does your study bring significance to maybe human health or human neurology?
So again, even in humans, our dopamine system plays a huge role in our light adaptation and the development of actual neuronal circuitry. And again, in diseases like Parkinson's, if you want to look at CNS in general. So we're just trying to get a better understanding of how that works, so we can kind of see what its implication is in diseases and those physiological processes.
All right. Thank you. Interesting study.
Thank you. So we've got a couple of minutes remaining. I'll just see if the audience has any questions. I'll give them 30 seconds to put any questions in the Q&A box. Okay, all good. Thank you. Thanks, Vrinda.
Thank you so much.
So this brings us to the end of today's session. Thank you to our judges, Dr. Barnaby Dixon and Darren Soniolu. Thank you also to our student presenters, and congratulations. So this recording will be made available online following the conference, and of course we hope to see you at our other conference sessions either later today or this week. Thank you all.
Thanks, guys. Thanks for having me.
Thanks, guys.
Great work.